Antibody, a water-soluble globular protein produced by lymphocytes. Antibodies are involved in the immune response by specifically recognizing and binding to antigens. Antigen, a foreign substance that elicits a specific immune response, such as antibody production. Bacteria, a single-celled organism with a varied morphology. Bacteria can have a symbiotic relationship with their host, or under certain conditions, bacteria can cause illness. B lymphocyte, a type of white blood cell or leukocyte that provides antibody-mediated immunity. When stimulated by a specific antigen, it gives rise to memory cells and antibody-producing plasma cells. Cytotoxic T lymphocyte, a lymphocyte involved in cell-mediated immunity. Cytotoxic T cells monitor the cells of the body and become activated by encountering cells with abnormal surface antigens. Once activated, the cytotoxic T cell destroys the abnormal cell. Macrophage, a large phagocytic cell that engulfs and destroys foreign material, damaged cells, and cellular debris. Another immunological function of the macrophage is to present antigens from foreign substances on its surface for lymphocyte recognition and stimulation. Mast cell, an immune cell found in the connective tissue that releases cytokines like histamine as part of an immune response to a local infection. Memory cell, a dormant lymphocyte that is cloned from the stimulation of a B lymphocyte. Once activated by a specific antigen, the memory cell undergoes clonal expansion to form more memory cells and antibody-producing plasma cells. Pathogen, a disease-causing agent such as a virus or bacterial microorganism. Plasma cell, a lymphocyte that is a clone from a stimulated B lymphocyte. Plasma cells are distinguished from other lymphocytes by an abundance of rough endoplasmic reticulum, indicating active antibody production. T helper lymphocyte, a lymphocyte that amplifies the normal immune response. When stimulated by an antigen-presenting cell, such as a macrophage, T helper cells release factors that stimulate proliferation of B lymphocytes and attract cells key to the immune response. Virus, one of the simplest pathogens capable of causing disease by invading cells. Most viruses have a simple structure consisting of a small nucleic acid genome within a protein shell. In order to determine blood type, we will visually centrifuge these blood samples to separate the blood components. The ABO blood group is based on the presence or absence of two major proteins in red blood cell membranes, agglutinogen A and agglutinogen B. A person can have A and B, or neither A nor B in his or her blood cell membrane. These blood types are referred to as A, B, AB, and O. If a person has type A blood, his or her blood will contain antibodies against agglutinogen B. These antibodies are called agglutinins. Conversely, a type B person will have agglutinins against agglutinogen A. Blood compatibility can be predicted by understanding the differences between the donor and recipient blood types. The donor's blood generally only contains red blood cells with no plasma. Therefore, it is agglutinins in the recipient's blood plasma that determine if the donor's blood will be compatible. For these reasons, type O is known as the universal donor, while type AB is known as the universal recipient. You may now test various combinations of blood compatibility by picking a donor and recipient and seeing whether the blood clumps. Cancer is the result of a group of closely related diseases. It can occur in many different tissues and is the result of changes in the cell that inactivate some of the normal control mechanisms within the cell. The first characteristic of cancer is anaplasia. Anaplasia describes the appearance of abnormalities in cellular structure. These cells can fail to develop specialized structures that are common to that cell. A second characteristic which is common to cancer is hyperplasia. Hyperplasia is the uncontrolled reproduction of cells, which can result in large masses of cells called tumors. Metastasis is the third major characteristic of cancer. Metastasis is a tendency for the cancer to spread into other tissues. The cancerous cells can detach from their cellular mass and may then be carried away to other parts of the body, where they can establish new cancerous growths. Malignant is the term used to describe this type of cancer. This characteristic is often used to distinguish a cancerous growth from a non-cancerous or benign growth. Acquired immune deficiency syndrome, AIDS, is caused by the human immunodeficiency virus, HIV-1, a virus with an RNA genome referred to as a retrovirus. HIV focuses its attack on T helper lymphocytes. When HIV infects a cell, it shuttles its RNA into the cytoplasm and quickly converts the RNA to DNA. The viral DNA inserts itself into the host cell DNA. Once in the host genome, the HIV DNA may lie dormant for several years. If the viral genes are activated by some unknown mechanism, the genes induce the production of new HIV viruses that can leave the host cell to attack and kill other T cells. As more and more T lymphocytes are selectively killed by the HIV virus, an immunodeficiency develops, AIDS, leaving the victim susceptible to a host of opportunistic infections. Virus, one of the simplest pathogens capable of causing disease by invading. Acquired immune deficiency syndrome. Vaccination is the process of exposing an individual to an antigen to stimulate an immune response and produce antibodies to that antigen. Vaccines employ the benefits of priming the immune system for exposure to a pathogen. Many vaccines are attenuated or disabled forms of a pathogen. These attenuated pathogens are recognized by lymphocytes, resulting in clonal expansion and production of plasma cells and memory cells. The memory cells lie dormant until later exposure to the virulent pathogen, at which point they become activated. This secondary response will involve contact with more immunospecific memory cells initially, which results in a faster, more robust immune response. Cytotoxic T lymphocytes are important monitors of our cellular immunity. This epithelial cell encounters an antigen. When this cell presents an abnormal antigen on its surface, it is subject to destruction by a cytotoxic T lymphocyte. Once an abnormal cell is identified, the cytotoxic T cell kills that cell to prevent infection of surrounding cells or uncontrolled proliferation of an abnormal cell, as in cancer. Fever is a nonspecific immune response to infection. Bacteria entering the body through a cut may cause an infection. Upon recognition of the bacterial invasion, lymphocytes release pyrogens that signal the brain to raise body temperature. An elevated body temperature creates an unfavorable environment for microbial growth. A fever also enhances the normal immune response. B lymphocytes are cells specialized to respond to antigens. B lymphocytes have surface antibodies that are antigen-specific. When the B cell binds with the antigen, the cell undergoes clonal expansion. Some of the progeny become plasma cells that act as antibody factories. Other progeny become memory cells that remain dormant until another exposure to this antigen. Inflammation is a nonspecific immune response to infection. Bacterial infection signals mast cells to release a chemical signal that causes increased blood flow and vessel dilation. This allows phagocytic cells to be released from the capillary into tissue. Phagocytic cells engulf and destroy bacteria. The infection is encased by a connective tissue sac. The remains of the phagocytic cells and fluid are exuded from the tissue as pus. When a cell is invaded by a virus, one of the mechanisms for a nonspecific defense is to protect surrounding cells from infection. Upon viral infection, interferon may be released from the infected cell. This initiates antiviral defense mechanisms in all cells that receive the signal. Although the virus replicates and the infected cell dies from the viral attack, protection of the surrounding cells helps avoid a widespread infection. Lymphatic vessels recycle excess tissue fluid that accumulates in the interstitium as a result of normal capillary leakage. The walls of the lymphatic vessels act as one-way valves, only letting fluid enter. Once in the vessels, the lymph is advanced toward the systemic circulation. Along the way, the lymph is filtered through several lymph nodes where it is inspected for foreign substances. Upon reaching the thoracic duct, the lymph re-enters circulation. Phagocytic cells are specialized to nonspecifically consume and digest antigens. The macrophage is an example of a phagocytic cell. In addition to this nonspecific cleanup function, macrophages digest engulfed antigens and place small antigenic pieces on their surface for presentation to lymphocytes. T helper cells are another type of lymphocyte that aid in the immune response to an antigen. When a B lymphocyte and a T helper cell both recognize the same antigen on an antigen-presenting cell, the T helper lymphocyte responds by producing factors that assist and amplify the B cell's clonal expansion. In addition, the T helper cell releases other factors that attract additional B lymphocytes and phagocytic cells. This is a histological image of a section through the lymph node. Lymphocytes play an important role in controlling the immune response. In this image from the intestine, you can see an accumulation of lymphocytes in the connective tissue of the lamina propria. This is a histological image of a section of the ileum. Notice the lymph node-like Peyer's patches relationship to the mucosa and submucosa. This is a histological image of a section through the spleen. This is a histological image of a section through the thymus.